Okay, hi, my name is Sanika.
My name is Yashvi.
I'm Anoushka.
And I'm Sid.
And our project is on Monastrol and its effect on colon cancer cells. So as a brief outline, first we're going to talk about the basics of cancer, then we're going to go into the background of our project, then we're going to talk about current research and our future plans.
So first, we're going to go over the origin of cancer and then mitosis in healthy versus cancerous cells. So cancer is a disease that is characterized by the uncontrollable growth of defective cells in an area of the body. So normally human cells grow and multiply through cell division to form new cells as the body needs them. However, sometimes this process breaks down, resulting in abnormal cells growing and multiplying when they really shouldn't be. These kinds of cells may form tumors, which can be cancerous, and non-cancerous, which is also called benign. Cancerous tumors spread or invade nearby tissues and can travel to other places of the body to form new tumors. This cancer can form either solid or blood cancers, and blood cancers can include things like leukemia. Benign tumors do not spread to other tissues and usually don't grow back when they are removed. However, they can grow quite large, which can pose a threat for certain parts of the body like the brain.
Okay, so now I'm going to be talking a bit about mitosis in healthy cells versus cancer cells, as a change in mitosis plays a key role in cancer. So to start out, molecules known as cyclin-dependent kinases regulate the cell cycle. They ensure that there's checkpoints between each stages to make sure that everything is going smoothly. However, in cancer cells, these checkpoints are overridden, which leads to the uncontrollable growth. In addition, there's genes such as the p53 gene that suppresses tumors and the RB protein that inhibits and controls the cell division, proving to be vital genes for our cells. However, cancer cells have mutations in these genes, which inhibit their true functions, thus aiding the cancer cells to divide uncontrollably. Lastly, kinesins are another important molecule, as our main research focuses on them. And there are molecules that are involved in functions such as microtubule dynamics, chromosome segregation, and spindle formation, which we'll get into later in our presentation.
So now we'll talk about the background. So in the background includes how colon cancer begins, a little bit about Monastrol, and induction of apoptosis by Monastrol. So first, how does colon cancer begin? So colon cancer is a type of cancer that's very aggressive, and if untreated, it'll move across the phases very quickly and cause a lot of issues. And so this is how colon cancer basically develops. So it starts as a very small hyper proliferation and eventually turns into the invasive cancers. So there's basically three different polyps. So a polyp is generally an area where there's uncontrollable cell growth, and that's what leads to the cancer. So the first polyp is called adenomous polyp, and so basically that is sometimes caused a cancer. And so whenever you're diagnosed with that, it's possible that you can get it treated very quickly in the early stages, like the hyperproliferation stage, and get it cured. Hyperplastic polyps are generally not precancers and haven't caused many issues. But the third type of polyp, SSP, is the most dangerous because it starts in the later stage. It's more visible in the severe dysplasia stage. And when it's in that stage, it's very hard to control because the normal cell growth already started and eventually takes over and becomes invasive cancer. So how do we treat this? Well, we thought about Monastrol. So what is Monastrol? Monastrol is part of a kinesin motor family. So essentially what it does, it moves around and goes around, and essentially what its function is, it arrests regular spindle growth cells. So what happens is whenever you're doing metaphase or anaphase in the mitosis stages, so the replication of replication, and whenever there's a spindle formation, there's sometimes irregular spindle formation when the chromosomes don't line up properly, and that's what can cause colon cancer and sometimes called irregular spindle fiber growth. And so what Monastrol basically does is goes in there as represented in the image on the bottom and fixes the chromosome structure and removes all issues to make sure that the replication goes smoothly and only destroys the target part, not the entire cell. So there's essentially three main parts of the Monastrol protein. First part is the motor, which highlights the ATP. So ATP is an important is important for Monastrol because ATP is used for movement, which is very important when it has to get to the chromosome quickly. The stalk and tail are for the interaction with the chromosome. So once it reaches there, how it reacts with the chromosome is using the stalk and the tail. And the combination of these three lead to apoptosis, which will be discussed later.
So now we're going to be getting into a bit of a current research surrounding Monastrol and its effect on cancer.
So EG5, also known as the kinesin spindle protein, is a motor protein involved in the proper establishment of a bipolar mitotic spindle. So Monastrol inhibits both the basal and microtubule stimulate ATPase activity of the EG5 motor domain. Monastrol partially inhibits the EG5 ATPase activity by binding directly to the motor domain. However, Monastrol does not interfere with the formation of the motor empty complex and is not competitive with ATP's binding to EG5, which is different from many other ATPase inhibitors. Monastrol appears to stabilize a conformation that allows for reversals at the ATP hydrolysis state. These interactions at the hydrolysis step alter the ability of EG5 to generate force. Therefore, the EG5 complex cannot establish or maintain a bipolar spindle.
So now we'll introduce a new aspect, which is tumor hypoxia and acidosis. So tumors in general thrive in microenvironments with limited access to nutrients and reduced oxygen supply. So acidosis is when there's an excess of acid, and tumor hypoxia is where the tumor cells have limited oxygen. So they both prevent anti-cancer drug efficiency, so it makes it trickier to treat the tumor. An assay by Cancer Cell International BMC uses different levels of pH to replicate tumor hypoxia and acidosis on HCT116 cells to test how efficient different clinical anti-cancer drugs are on the cells. So the pH most effective with the drugs was a pH of 6.8. And with testing other levels, they found out that the lower the pH is, or the more acidic, the worse it is on the effectiveness of the drugs. So these graphs shown are the different clinical drugs used on two experimental groups, which was the HCT116 at a pH of 7.4 and the AA HCT116 at a pH of 6.8. So as you can tell, the AA HCT cells did better overall with the higher viability. So using this assay, our adaptation will use their results in a different way to test colon cancer effects on the cells. So using slightly lower pHs, we will stimulate acidosis to prevent cancer therapy efficiency. And we can even adjust the levels in multiple trials to help support Monastrol.
So for the past 4 weeks, our team has been learning to grow our own HCT116 cells. We learned from Casey to ensure that we were working correctly and our cells were healthy. And we learned how to identify contamination and treat it accordingly, as well as how to fix any mistakes we may have made along the way.
Okay, so now we're going to be getting into a bit of our future plans. So one of the main assays that we plan on completing in the future is an MTT assay. The purpose of this assay is to measure cellular metabolic activity as an indicator of cell viability, proliferation, and cytotoxicity. The basic principle is that a yellow tetrazolium salt gets reduced to a purple formazine crystal by the cellular metabolic activity. The crystals are then dissolved using a solubilization solution, and the color is quantified by measuring the absorbance at 500 to 600 nanometers. The darker the solution is, the greater number of viable metabolically active cells there are. So we plan on using the MTT assay to determine the effectivity of Monastrol. We want to answer questions such as, to what extent does Monastrol help with cancer inhibition? How many cancer cells are led to apoptosis by Monastrol? And how the anti-proliferative properties of Monastrol lead to cell death. Through careful research, we have written a thorough protocol on using the MTT assay to demonstrate our cell viability and activity. However, the MTT assay is only the first step. By performing more assays, we'll be able to test the true effectiveness of the compound.
So after the MTT assay, we plan to perform other various assays and experiments to see how far we can take Monastrol and its effects. So we'll be doing a spindle formation assay to see how Monastrol actually affects spindle formation. We'll also be performing in silico docking of Monastrol derivatives with the goal to find the best derivative to inhibit the EG5 motor domain. And we'll also be applying Monastrol to our research about colon cancer to see if it can work as some sort of solution. So we'll be doing multiple variations of this, such as applying the acidosis/tumor hypoxia effect by lowering the pH to see the effects of colon cancer and altering the values of the pH to support Monastrol.
So this a cloud tweet. So we would like to thank ASDRP and the sponsors for helping us with this project. We'd like to thank our advisor, Ms. Ranganathan. We'd like to thank the lab technicians, especially Casey, who taught us about cell cultures and gave us the HCT strains. We'd like to thank Edwards Group for giving us the Monastrol and helping us make this project worthwhile. And we'd like to thank the group members and our parents for giving rides. Thank you.